In this lecture, we will discuss the evidence of evolution. There are 5 main bodies of evidence that support evolution: fossils, biogeography, embryology, comparative anatomy, and DNA and proteins. Each of these have subcategories as well, and some very specific descriptions, so make sure you're able to discuss each of these individually. The first one we'll talk about is fossils, and a fossil is preserved bone or other trace of an ancient organism. These are formed when dead organisms are buried by sediment. Other harder minerals slowly replace the calcium in the bones or in the shell of the organism, so we often see those bones that have been buried. We can also see something that the organism left behind that isn't necessarily part of the organism, like an eggshell or possibly a nest or a burrow or something of that nature. So anything that really shows that the organism was there. It doesn't necessarily have to be a bone or a shell. We can look at these fossils and see how organisms have slowly progressed through time. Organisms that are found in older fossils, things that are buried more deeply, can often show a step-by-step progression toward the more modern fossils and even modern organisms of today. So we see this slow progression through time when we line up these fossils in order. Paleontology is the study of these ancient items. We use morphology, which means that what an organism looks like, to determine more information about them. So here's a good example of a transitional fossil called the Archaeopteryx. This is this organism here, which basically would look like a dinosaur but with feathers. So you can see this is the head and the body here. These would be the arms or wings with some fingers there, and then the legs bent there. You can also see impressions of feathers in the stone surrounding the bones. So it's very clear that this organism was feathered, but also we can be sure of its size and its body shape. Unfortunately, very few organisms actually become fossils. It takes a very specific set of conditions in order for that to occur, so we have a relatively small number of these. Transitional fossils are anything that shows the transition from one species to another. So really every fossil is a transitional fossil. If you're going from left to right, can you pinpoint the exact place that the color stops being blue and then becomes red? No, you can't. There's purple in between. It varies depending on someone's perception, and there's not really one exact spot where you can say, yes, this is definitely red. Well, that's how it goes with evolution as well. There's no specific generation where a mother gives birth to a child of a different species. For most species, it's a very gradual change over thousands of years. So there's no missing link. All of the species are transitional. Here's another example of a transitional fossil that is very clear, and that is the Tiktaalik. You'll see this in a video that you watch later as well. This is basically a fish with legs that can start to crawl out of the water. Or horses have a very complete fossil record because they evolved quite quickly, and you can see the difference going from the smaller toed horses all the way up through the modern day horse. And all of these fossils are very easily distinguished and lined up accordingly. Another example is the whale. Pakicetus is a wolf-like creature that entered the water maybe due to ample food availability or to escape predators. And over time, those back legs became obsolete, so they don't use them anymore, and they weren't required. So there was an advantage of those organisms to not have the back legs and not spend energy to create them and have them drag them down as they are swimming. And we end up with organisms with very small or non-existent back legs. Whales of today still have a pelvis there to show where their back legs would have been had they had them. So those are fossils. Second is geography. And geography provides evidence of evolution because island species very closely resemble the nearest mainland species, but also they show variation from island to island based on the environment that you see there. So a good example of this is the Galapagos Islands and the finches that live there. They both greatly resemble the South American finches that are close to the mainland species. And then each individual island has different species of finches, which all are very similar in body shape and food source, but they have differences in things like beak size based on the types of seeds that are available to them. Third is developmental patterns in embryos, or embryology. This is using stages of development to determine relatedness. The closer the stages of development are, the more related the organisms are. And I have some photographs of embryos of 5 different species here. One of these is human. Can you tell definitively which of them is human? Probably not. Some of them have some specific characteristics, like the turtle. You could probably figure out which one is a turtle. That's this one right here. But each of these are so different that it's hard to tell what the organism, or each of these are so similar that it's hard to tell what the organism would be. And they all have gills, okay, right there, gill slits, and they all have tails here. But not all of these organisms are born with tails. Here's the organism that they're going to turn into. So this is a human embryo. Notice it has a tail and it has gills, but we're not born with those. This is a mouse, very similar looking to the human. This is a bat, a bird, and a turtle. So, in early development, human embryos and embryos of all other vertebrates are extremely similar. This is a drawing. It's not a photograph like the ones that I showed before. These are just, like I said, drawings of similar embryos. The fourth one is comparative anatomy, and so that's how we compare how organisms are put together and provide important evidence for evolution. So we use specific structures to show this. One of them is a homologous structure. And a homologous structure is one that is similar because it's a modified version of a structure that occurred in a common ancestor. So here is the ancestor of some winged insects. It looks like a dragonfly, but it's not. It's an ancestor to the dragonfly, and it gave rise to several different winged insect species. All of their wings are then homologous structures because they derive from the ancestor's wings. Another example is the bones in the front limbs of birds and dolphins. These are all homologous structures here because they derive from a common ancestor. So a dolphin, even though it just has a fin, it still has finger bones, hand bones, wrist bones, 2 little lower arm bones, and 1 upper arm bone, just as a dog, a human, and a bat all have those same bones. So these bones, even though they are not used individually, they're still found inside the dolphin's fin because it inherited them from an ancestor, the same ancestor that all of these organisms also descended from. This causes divergent evolution. So divergent evolution is, or homologous structures are found as a result of divergent evolution, and this is evolution towards different traits in closely related species. Analogous structures are those that look similar but evolve from different ancestors. So these are not evidence of relatedness, and it's a little bit confusing to distinguish between homologous and analogous structures. But homologous structures are those that do come from a common ancestor, and analogous structures do not come from a common ancestor. An example would be the wing of a bird and the wing of an insect. Even though they're both used to fly, they did not inherit those wings from the same ancestor. Analogous structures occur due to convergent evolution, where organisms have similar traits even though they are distantly related because they have similar selective pressures, like a shark and a dolphin. A shark is a fish, and a dolphin is a mammal, so they are very distantly related, but they have similar body shape, similar fin placement, and similar coloration because they both have evolved the best traits possible in the oceanic environment. Vestigial structures is our third type, and those are remnants of an organism's evolutionary past that have no function. For example, the wings of an ostrich. These are structures that are used for flight, but ostriches are not able to fly. So if they can't fly, then why do they need wings? Well, because they inherited them from an ancestor that used to be able to fly. Another example is the pelvis in a whale or the pelvis of a snake. The only function of a pelvis is to connect your hind limbs to your spinal column. Since snakes and whales don't have hind limbs, there is no purpose for them to have a pelvis. So then why do they have one? Because their ancestors had legs, and although they don't have legs when they are born any longer, they still have some remnants of the pelvis that is left internally. Here is an abbreviated list of some of the vestigial structures that humans have. Not all of these structures, this is not all of the vestigial structures, but it's some of them that I thought you might recognize. The last category of evidence is DNA in proteins, and this is typically a very convincing one, because you can see that the DNA sequence is an organism's history has been written in that sequence. And so we use a lot of compounds like myoglobin or hemoglobin or cytochrome C, things that many organisms have, so we can compare them across a large number of organisms. So if species have changed over time, then their genes should have changed as well. So the theory of evolution predicts that genes will accumulate more alterations in their nucleotide sequences over time. We can then create a molecular clock, which uses the differences between molecules like DNA or RNA or hemoglobin or cytochrome C to determine how closely related two groups are. So if humans and a rhesus monkey only have 8 differences, then they are more closely related than a human and a frog, which have 67 differences in the amino acid hemoglobin. The greater the difference, the higher the divergence, the higher the percentage difference in those compared molecules. So again, if we look at cytochrome C, which is one of the proteins in the electron transport chain of cellular respiration, the human and the yeast have a lot of differences, but a human and a pig have very few. That shows us that the human and the pig are more closely related than human and yeast. Therefore, closely related species will show more similarities than distantly related species. In most cases, evolutionary relationships indicated by DNA confirm those suggested by other areas. DNA evidence is relatively new. We haven't had the technology to be able to look at the DNA structures for a very long period of time. Darwin definitely wasn't able to do that. He was using these other types of evidence, and now looking back, the DNA and protein sequences confirm what he had originally thought about relationships between organisms. Some other specific gene sequences we need to bring up are pseudogenes. Pseudogenes are clues to a common ancestor because they are DNA that does not function. So humans have a ton of DNA that doesn't actually do anything. It really just takes up space, and it's in our DNA sequence, but we don't use it to build proteins. It's just there and gets cut out during protein synthesis. So then why do we have it? Well, because it's been passed down for millions of years from our ancestors. We might have genes that we don't use now, but that we do have in common with some other distantly related organisms. Hox genes also indicate a very distant common ancestor, and a Hox gene is what determines head-to-tail development in embryos. All genes are shared by organisms, so it's called a master gene. This is one of the criteria for being in the kingdom Animalia. Basically, it just tells where cells, based on their location, what they develop into. So if you're at the top, then you develop into the head and the brain, right? And if you're at the bottom, you develop into the extremities. If you're in the middle, then that's going to be the torso and the internal organs. So it's really just based on location in the very early stages of development. Insects have this, humans have this, anything in the kingdom Animalia has this gene to determine how our cells figure out what type of cell they're going to be. Before we finish up, I just want to get rid of a couple of misconceptions that you may have about evolution. One of them is regarding the origin of life. The origin of life is not at all addressed in the theory of evolution. Evolution is about what happens once life is already here, not about how life got here. There are several hypotheses to try and figure out how life originally began, but none of them are part of the theory of evolution. Also, humans did not evolve from monkeys. Nowhere in the theory of evolution is that even implied. What the theory of evolution states is that humans and monkeys share a common ancestor that was neither human nor monkey. But gorillas, humans, chimpanzees have all had equal amounts of time to evolve and share a common ancestor that is not any of those organisms themselves. Humans didn't evolve from monkeys because we're all modern organisms. Lastly, irreducible complexity is not something that occurs. Irreducible complexity is the idea that there are some things that are just too complex to evolve. Oftentimes the eye is stated as one of these things, but there are not any characteristics of humans or other organisms that there are no steps evolutionary to reach them. So it's possible to have half of an eye, a pigment cup here that you would see with some organisms where it is an indentation, and there's nerves that can determine if maybe it's just light or dark or some basic colors. Or a water-filled cavity here. This would be like the eye of a nautilus, where instead of having a lens over the top concealing it, it's just a water-filled cavity with a small hole and those optic nerves to capture some images. Even marine snails have kind of a step in between getting up to the octopus, which actually has a very complex eye. It's almost identical to the human eye, except for a octopus does not have a blind spot the way humans do. So even very complex things can still have steps in order to reach that level of complexity. And that is all for this lecture today. Please contact your instructor if you have any questions. Thank you.